But I'm very excited to have the next panel join the stage. I actually started working on the Bespoke Gene Therapy Consortium on my second day at the foundation and have been very deeply involved in its progress. So very excited to have them kick off our next session. Thank you.
Thank you, Courtney, for that introduction. And while we're getting set up here, our first speaker actually is going to be Dr. Peter Marks, the director of the Center for Biologics at the FDA. And his role in the BGTC has been really critical, and very happy to have him kick off the presentations. And then after he does, we'll go through an overview of the whole BGTC and how we're operating it currently. So, Peter?
So thanks very much. I'm going to tell you kind of how we ended up in part getting there. Just to kind of set up the story of why this collaboration was necessary. So let's see here. For those who aren't totally familiar with rare diseases, the reason why these are so important is because when you take all of the different rare diseases in aggregate, you end up having many diseases that may just affect even a few people, but you aggregate 7,000 plus diseases, and you have diseases that actually affect millions of individuals in this country alone, and many more globally. Additionally, when you think about rare diseases, when you start to think about intervening at the molecular level with common diseases and fixing genetic defects, you realize that what we're dealing with is a lot of rare diseases. And if we can get it right in the known rare diseases, we will be able to essentially extrapolate that to many other places. In this rare disease space, we have seen what gene therapy can do. Gene therapy has really had some clear successes in certain places, and one of its clearest successes in a disease called spinal muscular atrophy type 1, where this is a pretty severe disease which basically takes kids' lives by the age of about 3 years old. They don't make any of the developmental milestones that they should in terms of motor function, and eventually end up on ventilators and die. This particular gene therapy on the SEMNA gene, when given early in life within the first 6 months, it really transforms this disease. It may not be absolutely perfect, but you can see a child running around at age 3 is very different than no child at all at age 3. And so really quite a remarkable example of what gene therapy can do. Not all examples of gene therapy are going to be like this, because in many cases you won't be able to reverse decline, but in this case, if you get there before you've had decline, it's pretty remarkable what can be done. However, really there are some real challenges that we have in the gene therapy field, and particularly right now in the current economy, gene therapy is really at a critical juncture due to a combination of challenges with manufacturing, clinical development timelines which are challenging for people, particularly venture capitalists, to understand and to deal with, and different global regulatory requirements which have evolved over time, but which create very challenging situations for small products that have small patient populations. So at the Center for Biologics Evaluation and Research at FDA, we have been trying to address these things by advancing manufacturing technologies for cell and gene therapy through research and through collaboration through this FNIH Bespoke Gene Therapy Consortium, which we'll hear more about. Applying the platform technology provision, which was put into law this past year. Talking more about how to more clearly define the use of accelerated approval for gene therapy, working to explore concurrent regulatory submission in multiple jurisdictions around the world, and trying to apply some of what we've learned with the pandemic to rare disease gene therapy. And I'm just going to go through each of these relatively rapidly. The manufacturing issue here, and I show this all too often, our coffee cups here this morning look very much like this, so it's perfect. We do very well in the mid-size gene therapy production where the economics work so that you get good return on investment. Let's leave out the large-size cup for today. The problem is in the small cup where the setup costs for making gene therapy are such that we have not yet mastered how to make small batches of gene therapies for these small patient populations in a cost-effective manner. And that's really problematic because it's leading to both inhibition of research in the area and something that's really just a non-sustainable ecosystem. So how do we solve that? Well, we can harmonize manufacturing protocols, and I think, again, you're going to hear about that through the Bespoke Gene Therapy Consortium, because this is kind of one of the things that help bring this into being, and also thinking about automating manufacturing processes. That's something we could deal with on another day. The other issue that I think we really think that, and this actually is something that PJ has been talking about since before the platforms technologies provisions, is that really gene therapies represent something different than many of our other drugs, which is that the vector portion of an adeno-associated viral vector gene therapy, the vector itself is going to be the same often from product to product within a given serotype. And if we can find a way to leverage manufacturing information, toxicology information from one product to the next, we could save a lot of the rework that happens each time, and that may help bring forward more products more rapidly. And to wit, over the past couple of years, there's been a growing desire to see if we could do this and to put in place in law provisions that would allow us to have the ability to take an approved product and designate it a platform such that additional products could essentially leverage that approved product for the manufacturing and the toxicology, other information. So right now, this is something that's in law. We look forward to applying it. It applies to the products of a given sponsor. That's just because right now, manufacturing is so specific to a given individual sponsor that it's hard to extrapolate across programs. That may come in the future. The next issue is that in a lot of the rare diseases that we deal with, it's very hard to get long-term natural histories for these diseases. And it's also very hard sometimes to get endpoints that take long periods of time, years to assess. As I've mentioned, venture capitalists have short attention spans. They're looking to see these products progress. Frankly, their patients' parents are actually very well aligned with venture capitalists because they want, patients and their families would like to see cures or at least treatments in a more timely fashion. And one way to get there is to leverage accelerated approval. Accelerated approval is the provision that we have at FDA that allows one to use a biomarker or intermediate endpoint that is reasonably likely to predict an outcome to give an approval that then it becomes a full approval when we get the clinical data that confirms the accelerated approval. And gene therapies actually are really well suited to potentially use accelerated approval because oftentimes we know, right, we know the gene of interest, and if we can, particularly if we can measure that gene product in the tissue of interest, then we can use our accelerated approval provisions by leveraging animal models or human observations to do an accelerated approval. Now, human observations, how does that work? Many of the rare diseases have situations where there are individuals who are deficient in a protein, but they are not visibly affected. And so the idea here is if you can show that with a gene therapy you can get to a similar level in an affected individual, again, it's reasonably likely to predict and allows us to use these accelerated approval provisions. Another issue that I mentioned, this issue of global divergence in regulatory environments. You can imagine that if you have 25 or 50 individuals with a genetic disease in the United States, that's a small population. But it means that probably there's also another 50 to 75 individuals in European Union countries that have this, and might be equally numbered to both of those in the rest of the world. And so if we could find a way to make it easier for sponsors to submit their applications globally, or at least across high-income countries, we might find ways to take commercially non-viable entities right now and make them viable. Just as a benchmark, right now, if you have about 100 systemic treatments of a gene therapy a year, you're in the commercial viability range. We could argue over whether it's a little more or a little less, but that's about where we need to get to. So if we can drive more things to be commercially viable by having this kind of regulatory collaboration, it would make a big deal. And what we have done over the past few years is set up what we need to do a pilot where we will try to work with our European and some other colleagues to see if we can review applications together, potentially get to a decision on these together, which would then allow in the future, hopefully, these kind of shared submissions. The idea is also to harmonize on what our requirements are so that we don't have to have rework, which is one of the problems that has really been there for sponsors to date. And then finally, one of the issues from the pandemic that we learned, and it's something that we probably should have known all along, is that for rare diseases, we could really benefit from more communication with sponsors. And we learned that from the vaccine case, where granted vaccines weren't for small populations, they were very large populations, but the way that Operation Warp Speed expedited vaccine development was in part by having certain just mechanical things, manufacturing at risk, et cetera. But in part, it was by having interaction between the product developers and FDA on an ongoing basis, so that rather than having meetings that were punctuated at times during development, which often take somewhere between 30 and 75 days to schedule, the communication happened by email or telephone or Zoom on an ongoing basis. That really significantly sped up development of vaccines. The question is, well, wouldn't that be a good thing for small companies where the time waiting for responses from FDA is basically time that they're burning cash without actually advancing development? And so we are working to have a pilot. We'll be doing 3 products in the Center for Biologics. There'll be 3 products in the neurologic space for the Center for Drugs, where in this rare disease space, we will give sponsors, once these are chosen, and we're accepting applications now, we'll be making the choice and then announcing that in May. These pilot programs will get kind of Operation Warp Speed-like service in terms of communication with a project manager, and then we'll be seeing whether we make developmental milestones more rapidly, and also getting feedback from the companies to see if that makes a difference. So, okay, I've spoken for about 10 or so minutes about these things that aren't relevant to the AMP collaboration. So what is? Really, you know, we have the entity, the Center for Biologics Evaluation Research, and there's the AMP Bespoke Gene Therapy Consortium, which was set up. And when you actually look at it, a lot of our goals, with perhaps this small rim, perhaps of some of the global regulatory work we do, and some of the scientific work, but most of this really overlaps very nicely. So I think what you'll hear about is really what we can do. This collaboration may make a really big difference in this by putting together some of the essential elements that we need. Ultimately, you know, we work together, and hopefully in collaboration with Bespoke Gene Therapy Consortium, we aim at the center to really try to make 2024 a breakout year for gene therapies, hopefully setting up the way for a wave of approvals for these smaller gene therapies. And I will stop there, and thanks very much.
Thanks. So yes, I'll be talking about the BGTC here, and just very happy to say that it's an honor to be part of the leadership of this program, along with Timothy Miller, the co-chair here from Thermo Fisher Scientific, and also Peter Marks, who you just heard from. I really want to give a shout out to Peter, because I think without his commitment to this program and vision, I really don't think it could have worked. So we're very fortunate to have his involvement and his team, including Gopa Reshadori, who's here. And also our FNIH colleagues. Courtney mentioned she was part of this from the beginning, and Kira and Brad, who are wonderful to work with. So the presenters today, I'll be presenting along with Timothy Miller and Jean Didashti from NIH, who will be talking about the kind of the overview of the program, and then our colleagues from IQVIA, who are overseeing the BGTC Coordination Center, Amrita Jaishankar, Carmen Sivakumaran, and Jenny Pham. And so the way we'll kind of do it is Tim and Jean and I will give a BGTC overview, and then we'll turn it over to the IQVIA folks to talk about the operations and where we're going, and if hopefully there'll be time for some Q&A. So as Peter alluded to, we are really addressing a very significant unmet need. There are now an estimated greater than 10,000 rare diseases that we know of, and the number keeps going up, affecting over 30 million Americans. And that is a real challenge. From the standpoint of therapy, though, the good news about monogenic disease is that we know what the problem is, which is defective copies of a single gene, and we know what a therapeutic solution is, which is to put a working copy of that gene into the relevant cell types at the appropriate time in disease progression. If we can do that, we could either treat, prevent, or even cure diseases. And adeno-associated virus, or AAV, is a way to do that. It's really a delivery vehicle for therapeutic genes. In the wild-type AAV is a small virus that many of us have been exposed to that does not by itself cause any human disease. It has a very small and simple genome that can be simply removed and replaced with therapeutic genomes to create therapies. And looking at that diagram, and in principle it all seems very easy. So you might be thinking, well, what's the problem? Well, one of the problems is that while AAV in the wild type, if you're infected with it, doesn't cause a disease, in AAV gene therapy, we might be dosing people with 10 to the 15th of these AAVs over the course of a couple hours, and that does potentially cause some toxicity that has to be managed. Producing these drugs is actually very expensive and complicated, and the regulatory path is also very expensive, complicated, and complex and time-consuming. And the other problem that really kicks in here is that many of these diseases are so rare that while they could benefit tremendously from AAV gene therapy, they have no commercial interest. And then what happens is that some of the parents of some of these children have to essentially become AAV gene therapy drug developers. And you'll hear from one of those parents later in the day. So this is the challenge, and then that led to the setting up of the BGTC. And this challenge not only defines the need, but by focusing on, in particular, diseases that are of no commercial interest, we essentially predefine the pre-competitive space that we're all able to work in under this AMP program. So our goals, as was stated, was to improve AAV vector production and target gene expression, essentially to make the drug work better when it gets into people, streamline preclinical and product testing, and to kind of harmonize and validate the minimum sets of manufacturing and preclinical testing requirements while at the same time maintaining patient safety. There are not actually, at current time, minimal sets or even completely agreed upon sets of assays for assessing whether these drugs are safe enough to go into people. A big focus of the BGT is to streamline the navigation of the regulatory pathway and product development for AAV gene therapies. We sometimes talk about streamlining the regulatory path, but I want to be very clear that's a kind of shorthand. The regulatory path is set by the FDA and is enabled in statute. What we're trying to do is help people navigate that regulatory path, go through it more easily and more efficiently. By developing things like a standard regulatory submission package templates that everybody could use. And also, very importantly, we have developed the BGTC regulatory playbook that helps people to go through this regulatory process. And I'm happy to be able to announce that the very first version of this regulatory playbook actually went public today at 10 a.m. So after this symposium is over, you can all go take a look at it. And ultimately, what we'd like to do through this is to change the consideration of what is a disease of commercial interest. If we can kind of lower the bar in terms of the entry to get into the clinic, we can change what people think about as disease of commercial interest, and ultimately hope to deliver the problem of gene therapy to all the patients and families who could benefit from it. And so we have a very broad set of public and private partners. Many of the different parts of the NIH, as you can see there, are involved. 11 institutes and centers at the NIH, many different public partners who are here, private partners. Particularly want to give an acknowledgement to the California Institute for Regenerative Medicine, or CIRM. They are partnering with us in a rather novel and creative sort of partnership. It actually allowed us to bring more diseases into the BGTC than we had been planning to do at the beginning. So very much appreciate that. And you can see the total funding there roughly balanced between the public and private contributions. And so the BGTC is really composed of two parts. There's a basic biology part that's focused on understanding the basic biology of AAV as it relates to making it easier to generate vectors and enhancing therapeutic gene expression in patients. The larger part of it, though, is the clinical program that's focused on streamlining the navigation of this regulatory path, going from the idea of a gene therapy to the first-in-human patients. And a major focus, again, is standardized minimum packages of critical quality assets and GLP toxicology studies that we hope will streamline the navigation of that regulatory path. And I'll just want to mention here, because they're not present at the meeting, but some of the awards that we've given in the AAV biology research. We did 2 for enhancing vector generation, and the remaining 6 are for enhancing therapeutic gene expression. It was honestly a bit of a challenge to do this in the pre-competitive space, because there's a great interest in amongst companies to do this, but we got some good applications that we're funding, and look forward to that and seeing the results. And I'd also note that this is an international program open to investigators not only in the United States but throughout the world. And so I'm now going to hand it over to Timothy Miller to talk about the clinical program and the choice of diseases.
Thank you, PJ. So when we began this work, actually it's been quite a while since we did. We started in a number of different nominations that were received from different groups, both patient advocacy organizations, academic groups, and others. And this was an open submission process, and we received 62 different nominations. There was a multifunctional group that were made up of experts from various fields that came together and reviewed all of these. We sorted them down to what we thought would be 14 candidates that were selected on a number of criteria. Of course, some very specific criteria, that they were monogenic disorders, and that there was not any significant commercial activity already ongoing within the area. And then we finally selected 8 of them, and I think we announced that last year at ASGCT. So we have been continually moving forward on those, and we're going to walk through a few of those today. But of course, that means we also are working to set up not only the initial clinical discussions and but all of the preclinical work and the manufacturing for those different programs. So we've separated them out here into 3 different organ systems, 3 in the ocular, 3 in neurologic, and 2 in the more systemic diseases, although many of these have some multisystemic dysfunction or impacts throughout the disease. If you haven't had a chance to look at this before, you'll also see now that we're talking about 3 different routes of administration, which gives us the ability to have experience in and some experience working with the process of the clinical trial development, the preclinical work, and the regulatory pathway with these folks. We have separated them out a little bit more if you don't know these diseases. I'm a neurologist. I've seen patients with several of these diseases, but not all of them. But as you can see, the ocular diseases are using both AAV8 and AAV5 vectors, and you can see that the route of administration is both intracorneal in the first and then a subretinal injection in the latter 2. So we do have experience across them. All of these are significant illnesses, and I believe there were posters on a few of these yesterday that you might have had a chance to see in the poster session. In the neurologic categories, 2 of these are clear enzyme deficiency disorders, but obviously they are also using the same vector across, but different levels of neurologic route of administration, intracisterna magna for the MSD program, and the other 2 are noted as intrathecal. Each of those will probably require some additional delivery mechanisms or some explanation of that, but very exciting programs that we have early scientific data in that we think it has a good chance of moving forward. And then finally, are in 2 systemic conditions, propionic acidemia and also MPS type 4A, very significant unmet medical need in all of the populations of these diseases, but providing us an opportunity to really deliver some of the promise of gene therapy in these very rare populations. And I think I'll pass it to Gene Tedashti.
Thanks, Tim. Sorry, there we go. So I'm Jean Tedashti. I'm a program officer within NIH, National Center for Advancing Translational Sciences, and I work very closely with Dr. P.J. Brooks and everyone on this panel in supporting the BGTC. What I'd like to focus on today is the Coordination Center. As Tim nicely outlined, we have currently 8 projects within our portfolio. 5 of them are supported through the consortium and NIH funding, and the remaining 3 are funded in part through the BGTC and in part through SARM funding. So we established the Coordination Center as sort of a hub to move these projects through their clinical programs with the goal of reaching first-in-human studies and onward. The Coordination Center was established through a government contract. From the time that we actually awarded the projects through October 2023, we worked on the SOW and we executed the contract, and the primary contractor is Black Canyon Consulting. I'm not sure if they're in the house today, and IQVIA is the subcontractor. The Coordination Center consists of a large program management operation component. There are 4 cores: the preclinical core, clinical core, regulatory quality assurance, and data management and analysis core. And you'll hear more about them once IQVIA gets up to talk. I'm not going to go into details, but it's just to show you the breadth of support for each of the cores and then the program management operation component of that. So this illustration is to show you the structure of the BGTC as a whole, the governance, and outlining how we've set up the teams for each of these given projects within our portfolio. So at the top of the slide, what you're looking at is really the infrastructure that was put in place within the past 2 years. Prior to the coordination center being established, we have our steering committee. I think that's consistent across all the AMP projects. And then we created what we're calling sub-teams for each of the core disciplines, so manufacturing, preclinical, clinical, regulatory. And these have been outstanding consortium members that contribute their expertise in these given areas. We also have a sustainability strategy working group. This advisory group is forward-thinking, projecting down the line as we move these clinical programs forward. In the middle of the slide, you see it's supposed to be a card deck illustration for the 8 projects within the portfolio. In the center, we have the BGTC Coordination Center outlined. As I described, it's the PMO component. We have our 4 cores. And the idea is that the BGTC Coordination Center will also help to run our centralized testing. We envision centralized testing from a product perspective for critical quality attributes, preclinical testing that we're going to be conducting for a lot of these projects, and then also patient testing, for instance, antibody titer testing. Another part of the cross-functional, by the way, what I failed to mention is that the card deck represents really our cross-functional team. For each of our projects, we have established this cross-functional team. So the coordination center sits across all the cross-functional teams. What's unique to each of the cross-functional teams is the sponsor representation. NIH is planned, NIH NCATS is projected to be the sponsor for 5 of the projects within the portfolio. And then for the CIRM-funded programs, the PI or the entity that holds the CIRM grant will be the sponsor for those. The PI and their team are represented, of course, in the cross-functional team. Their expertise is very important. And what's really important to us are the patient advocacy representatives that sit on these cross-functional teams, and they've been a part of the team from inception. And also, I don't want to forget to mention that within our cross-functional teams, we have representation from each of our sub-team members, so that we make sure that their expertise are included within the cross-functional team and across the portfolio. We really look at our sub-team members within the consortium and the expertise they provide as our advisory body. We use them quite frequently to address whether it's a manufacturing-related question, a preclinical development-related question. It's sort of endless. So they have a very significant role. And then, of course, the coordination center is our implementing body and reporting any outcomes. And this is a cyclic process. So needless to say, we have a lot of meetings and a lot of interactions, and we work very closely together. So now I want to highlight what PJ touched on, which is our major deliverable that we're excited about that was just released today. I think, if I'm correct, you can use the QR code on the screen, and you can look at it now or later. This is what we're calling version 1.0. So really, version 1.0 of our playbook serves as initial framework. And by the way, this document is best practices for gene therapy product development. And as we move forward with future iterations of this playbook, we are going to incorporate what we consider minimum standard packages with respect to pharmacology, toxicology, with respect to manufacturing attributes, critical quality attributes. This will all be incorporated in future iterations and our learnings from that. And I'd just like to give a big shout out on behalf of the BGTC leadership For all the BGTC members, the consortium members, the contractors, everyone has really put forth a lot of effort to get, for the past 2 years, and also to get these projects up and running, and we'll continue to do so. We really do work closely together as a team. So with that, I'll hand it over to Amaretha. Sorry, I'm going to butcher her last name. I'll let her introduce herself.
Thank you. Thanks so much, Jean, and thank you all for being here. On behalf of our IQVIA team here, I just want to say thank you so much. We're so delighted and honored to be supporting this transformative initiative, and thanks to the BGTC leadership and to the incredible asset teams. I see a couple of them here, so thanks for coming out. So I'm just going to kick things off here before I pass it to Carmen and Jenny, who have been leading our project management and regulatory work with this initiative. You've heard about the program overview already, and we'll be stepping in to discuss the challenges that the BGTC initiative is addressing and the key features of this coordination center and our early activities and outputs. So you've heard about a number of amazing collaborative initiatives over the last couple of days, and BGTC is clearly a really exciting initiative that is unique in that it simultaneously addresses multiple key needs in our industry. So we have a mandate to include patient group engagement throughout the developmental process. You've heard that there are 8 assets team, each at a different stage with unique needs, multiple sites, mixed IND holders. So supporting IND holders to fulfill their regulatory responsibilities, helping navigate the regulatory pathway, and incorporating this really broad multidisciplinary expertise across the government and industry that Jean already spoke about, has created this unique structure for BGTC where we really hope that when we come back here in 5 years, you'll see that all of these assets are in human already, and we hope that this would create a roadmap and a model that can be translated to other therapeutic modalities and indications as well. So in designing the right solution, several key questions needed to be considered. We have these multiple asset teams, each having different needs. How do we meet the needs of each of them and meet them where they are? How do we bring these common findings across all of the various assets and create that learning at a program level and create synergies? How do we manage communication and feedback among all of these stakeholders that we've discussed? And so each of these sort of questions led to a work stream within the coordination center that my team's going to discuss as we move through this presentation. And obviously there were considerations of data management and analysis, quality management systems, long-term follow-up, which is such a key need in our industry. It's a huge burden for individual sponsors. How do we reduce the patient burden? How do we create these centralized solutions? Is also a work stream. And then, we heard about the playbook. So how do we help navigate the regulatory pathway and accelerate development of these gene therapies for rare disease? And I saw multiple phones go up and that QR code came up. So we're really excited that this is public and look forward to feedback. And with that, I'm going to hand it over to Carmen to discuss each of these work streams that we've developed for the BGTC Coordination Center. Thanks.
Thank you so much once again to the organizers of the AMP Symposium and to the BGTC for having us. It's hard to follow such a star-studded cast here, but I'm thrilled to share a little bit of insight into the component tree that makes up the Coordination Center and how we're accomplishing this. So a large part of the early activities within the BGTC Coordination Center was on engaging with the cross-functional project teams to assess their readiness for their next milestone. And so what we did here is we conducted detailed assessments of all of the team's materials that they had available to share to really understand where each of these 7 project teams across these 8 assets were in their development pathway. And we did this through a series of meetings as we reviewed their materials. We did this in 2 stages. We had an introductory meeting to level set, expectations, let them know what the process was, introduce ourselves, and then a more in-depth kickoff meeting where we had the patient advocates, our BGTC sub-team representatives. And then for some teams, we had some smaller, more focused meetings to really dive deeper on key topics such as data management or manufacturing. And the main output here was what we're calling readiness assessments, which will lead to formalize go-forward strategies for each team. And so a readiness assessment is really a comprehensive, consolidated summary of our observations as we analyze the materials, with detailed sections across each of these disciplines, and a synthesis of what their next steps should be as a team to meet their next milestone. And part of this plan is having each team have a defined project plan. And what we'll do as the coordination center is we will have a master Gantt chart to have a consistent, coherent, and visible way to monitor and track all of the project's progress as they move through their development journey. And of course, these are all consistent with the BGTC approach, the content in the playbook, and we'll repeat this type of assessment with new materials and as they go through each milestone over the course of the program. So as we engage with these 8 assets through the readiness assessment process, we found expectedly that the project teams really are on a spectrum of levels of preparedness for their next milestone. So we've compiled some early learnings here, and I'll just highlight a couple here. One thing that really stood out to us, really on the data management front, was that most teams have a natural history study planned or underway. And so an important feature might be to assess the robustness of their systems and their current plans for use of that data in clinical trials. Another key finding for us was on the program level, with such a complex program with different stakeholders, there are a lot of questions about the roles and responsibilities from the PIs of these project teams, for example. And so what we did and what we're continuing to refine is a DARE framework, which is really an alternative to a RACI that's more focused on decision-making, with DARE standing for Decider, Advisor, Recommender, and Executor. And we hope this will also help clarify the roles for each of these teams. And another thing that really stood out to us from yesterday's fireside chat with Francis and David was that when the question came up about what they would have done differently looking back, the first thing that was said was they would have involved patients from the very beginning. And so we really relish the opportunity that we have here to learn from the past AMP programs, and we have the patient advocate representatives as part of each cross-functional team. But we also note the importance and the value of having a programmatic PAG engagement strategy as well, to really leverage the full potential of our involvement with the patient advocates and ensure that we're engaging with each of these groups in a fit-for-purpose way as we go along. So I'll also share some learnings into what the drivers could be for each of these asset timelines. And what we have here is a snapshot of what we presented to the steering committee. And what it was meant to be was really a stake in the ground that we can continue debating and discussing as we continue to refine and iterate on our thinking for what the accelerants could be for the program. And what we learned from the discussion is that there may be more flexibility than we thought that we could actually take advantage of in the context of rare disease. For example, for the first immutable driver on the left, one of the points of discussion was that actually INDs can be filed with a batch that's pending, but with a clearly articulated plan. So, while we continue to refine what we think might be accelerants across the program, we also note that there's a lot of variability, and therefore also many opportunities for harmonization to reduce that variability and reduce the uncertainty that comes with it. And what we're seeing, which of course was the goal from the beginning, is that across the development pathway for these assets, we see these one-off asset observations that really point to potential accelerators at the program level. And so you may see things come up once or twice, but then once they come up a few times, they tend to become a trend. For example, similarities in manufacturers and vendors point to efficiencies that we can draw on for process qualification and testing. Similarities in animal models, we can harness for accelerators in terms of minimum requirements for study design. Similar project teams having similar needs for an EDC for natural history studies, we can leverage for our design for a centralized data management approach. We're continuing to refine our approach across the program, and we are continuing to learn from these teams as we engage with them. So as we continue with these project teams on their journey, what we will continue to do is manage and communicate their status across the program to the leadership, through a dashboard such as this. So we have the assets listed according to Oregon system, with some key milestones and what we expect for the status. And we'll be continuing to monitor and report on this and continue to support these teams as necessary. So I'll move on to the next component for the Coordination Center. And as I noted, data management emerged as a nearer-term need for many of these teams. And so we're continuing to define the strategy and approach for a centralized data ecosystem. And so this, we hope, will enable continuity between the different data sources, notably in natural history study, clinical trial, and long-term follow-up, facilitate sponsor oversight through centralized data management and quality planning, power integrated data analysis within, across, and between the project teams. And I know this hopefully resonates with a lot of the data-focused presentations that we had today and yesterday as well. And the first instance of this data solution we hope to get going this year. And we hope to also continue translating those learnings across the portfolio from the first asset through to the next assets to come after. And another key component with sponsor responsibilities is, of course, quality management. And what we really are looking to do here with a QMS model within the BGTC is have a defined framework for how sponsors within the program are able to ensure accountability and regulatory compliance and quality as part of their obligations. So this is nothing new here, it's industry standards, but really doing it across the 8 programs, it would be new for us. So it's the application within this context that is new. And so our QMS model would establish a standards index, description of regulatory and quality documents, enable sponsor decision-making, and ensure the development of the right documentation, especially in the bespoke nature of this program. And of course, this is probably more intuitive for our colleagues in industry here, but not very much for our project teams. And what we're hoping to establish with this QMS is really the establishing the structure and the communications for quality. With at the QC level, you have your vendors, CDMOs, CROs, clinical sites, communicating and executing on the QC, ensuring that the products meet the quality standards with their processes, structure, contracts, documentation. You have your QA level with your quality auditors, ensuring that safety and standards and quality are met by the QC. And the QMS level where we have the sponsor defining the structure with the reporting and sign-off for the overall system. And so how these levels across GMP, GLP, and GCP get wired together needs to be defined before the first study starts, so it's one of our near-term priorities as well as part of this program. All right, and then of course, long-term follow-up, we're dealing with AAV gene therapy, so long-term follow-up is always top of mind with the current FDA guidance of 5 years after dosing. All project teams, of course, have similar priorities about reducing patient burden, ensuring patient retention, and closing gaps in data as they continue following patients. And we might have some variations in the type of patient support that's needed with the unique needs of each asset, but overall, the concerns and the priorities in data collection are quite similar. So what this really was clear to us, that this is really an opportunity for a common long-term follow-up framework, where from our early assessments of the assets, we noticed that all the assets have an inflection point around year 2 or 3, where they tend to drop off and follow-up reduces to annual visits. And most of the protocols also conclude the study period for endpoint analysis around that time as well. So there's really this opportunity to have a synergistic long-term follow-up program across the assets, and as all assets move to decreasing follow-up touch points, look at remote monitoring opportunities, look at moving things into the community, and these are some of the study design questions that we would think about as we continue exploring this framework. Where the opportunities are to harmonize, if there can be a central PI model, especially among the assets within the same organ system. So more to come as these assets get further into clinical development. And last but not least, the playbook is the first public output of the BGTC program. So we are very excited, so I'll say for the third time, that it is live today. So looking forward to getting your feedback and having everyone take a look at the playbook. We're very excited. The playbook will incorporate the BGTC learnings and continue to evolve with the program, and will act as a one-stop shop for acting as a simplified roadmap with templates, with guidance, to streamline the product development and navigation of the regulatory pathway. And really what this graphic here is meant to convey is that there are many ways up the mountain, towards the summit of getting therapies into patients, but the playbook is really meant to be a guide, your handbook, to help you along the way, to show you a simplified roadmap to optimize your success in getting therapies into patients. And with that, I'll conclude before handing over to my colleague Jenny, which is a recap of where we're going. So I told you a little bit about what our recent activities were with current project team engagement activities and readiness assessments. We'll be continuing to design and look towards implementation of our centralized DMA approach as we move into steady state engagement with these project teams. Of course, our QMS program and framework is our next priority as well as we continue tracking and reporting on the progress. And then, of course, continuing to support these teams on wherever they need us, contingency and continuation planning, sustainability with long-term follow-up, and continuing to centralize the capture of learnings with the playbook. In parallel with these activities, we define a feedback loop with the consortium where we continue to bring their expertise in. We're developing a toolkit for the project teams that can be applicable to the broader community with templates and resources, and we're continuing to define a series of best practices for a platform approach. And of course, always keeping our eye on the outcome here, getting our BGTC portfolio assets to IND in their first in-human trial, defining a roadmap for similar developers in rare disease, and hopefully defining a model that's extensible to other therapeutic areas and modalities. So with that, I thank you, and I'll like to open the floor to questions, and I'll hand over to Jenny for a Q&A for the rest of this session. Thank you.
I'll wait here until there's questions.
Okay.
I shall just stay.
Okay, thank you so much. This is truly a road less traveled. So at the Dental and Craniofacial Research, we deal with a large portfolio of craniofacial disorders. And if you looked at the human spectrum of inherited disorders, 50% of them actually involve the craniofacial complex. However, intervention has to be during development in utero. And I know this is very futuristic, but I wanted you to forecast any possibilities that we can actually deliver within the amniotic cavity, either a small molecule replacement therapy or an AAV gene therapy approach. These conditions actually quite devastating. There's only surgery that is the option right now, and it's normally performed on very young infants, and it requires lifelong management. Be nice to hear your thoughts.
I can take that one. I know that for some lysosomal storage disorders, there's already been work going on delivering enzyme replacement for those in utero. And as part of another program we have at NIH, the somatic cell genome editing program, we do have one project that's looking at doing delivery of genome editing in utero. So I think it is a possibility. That's about the best I can answer.
Thank you. The German government actually approved of a therapy for X-linked hypohidrotic ectodermal dysplasia, which is a rare condition, and it was successful on just one pregnancy, of course. But
okay. So this is tremendous. I'm a geneticist. I really appreciate all this progress. I think the 8 cases are well chosen. It was interesting to go through the posters as well as your presentation. But it triggers for me, and I'm sure for many others, especially lay people, what would be next? Are the other highly rated proposals out of the 62 that were not in the final 8 likely to be reconsidered at a subsequent stage? Is there going to be a whole new call for proposals and time-consuming process? At what stage in the 5 years do you think you might have in the detailed time chart you've already presented enough information to consider how to expand the program, assuming it's going well?
Yeah, I guess I can try to answer that one. We don't plan to incorporate any other diseases into the program. We really, the timing is such that, quite honestly, we'll be quite lucky to get all of them done in the 5 or 6 years that we have. We certainly hope that the work that we do, though, through the playbook and making the information available will benefit many, many other diseases as they go through the regulatory path. So it's really designed, as we said, to help navigate the regulatory path rather than to develop AAV gene therapies for a huge number of diseases. We certainly are familiar with many of the ones that applied and were not accepted. That was a very difficult decision for the team, the clinical team. You know, we've done what we can to try to help them to find other sources of funding through within the NIH or CIRM or other funders.
Great. Well, it's terrific. Good.
Thanks for a great overview, Virginia Savova-Sanofi. My question is, what is exactly the role of industry in this consortium? Because you mentioned there is industry in the steering committee. Do you want other partners? Are you getting involved? And then kind of in the same vein, right, there's obviously gene therapy approaches that are being developed in industry for diseases that are perhaps not as rare as the ones you're focusing on, but still could benefit from the approach that you're defining and also from some of the preclinical studies perhaps that you're doing, because something that you must be addressing in those studies is how the AAV capsids themselves are behaving. And as you pointed out, it's basically a capsid with a payload. So if you know something about where the capsid is going and what it's doing, that could help other programs, including not the ones you're focusing on. So what is your take on essentially broadening the impact of the wonderful work you're doing?
So I'll start with your first part of your question. Our industry partners have been tremendous. I don't know if you remember the image that I showed, but really the expertise that our industry partners, as well as government, we have representation from the NIH on our steering committee and our sub-team members. The expertise that they bring to the table, their experience with route of administration, the different serotypes that are within our profile have been tremendous. Several of them have had successful IND filings with the agency and even marketed gene therapy products. So their experience gained through whether it's preclinical study, trial design, particularly for these small patient populations, has been tremendous. So that's really, it's the expertise that they bring to the table and how they're helping our projects move forward is how we're utilizing them. Our advisory committee is what I'm calling. And PJ, I'll turn it to you.
Yeah, I'll just say too that I think if by making the information public about how to more efficiently navigate the regulatory path, that will benefit many companies as well. And as I said, we hope that by doing that we can kind of lower the bar to what constitutes a disease of commercial interest to get more companies trying to commercialize AAV gene therapies. So that's really how we envision it.
Just to clarify, because maybe I didn't catch it, do you currently have people that are actually currently working in this industry on your steering committee?
Yes.
Maybe I didn't understand.
Yes,
Okay,
yeah.
Penny Dax, AFTD. Thank you. What an exceptional program. Just how this could scale to so many different rare diseases that have nobody or no path forward. So thank you for your work. I was very curious. We also have, while most rare diseases with genetic origin are pediatric, you have quite a few adult onset degenerative diseases. I represent FTD, which is an adult onset with quite a few different rare genetic causes amongst that population. There's also Huntington's, there's ALS, there's others. Do you envision any nuance, shall we say, in the playbook, as well as some of the, just in general, the guidance of when you're having an adult population that you're treating with different biology, as well as different risk factors and governance and compliance issues compared to the pediatric populations?
What I can say is that some of the asset teams do have a target population, pediatric, and so we're looking at understanding the direct-to-patient prospective direct benefit for pediatrics and understanding if there are adults in those populations that would make sense to run some of the safety studies, early safety studies on them to understand safety for the pediatric population. And those discussions would be, or those proposals would be presented to the FDA in a pre-IND meeting, for example, if there is a logical reason for that and there are no ethical concerns with that. And then those learnings would, I think, would be incorporated into the playbook. Now, how that relates to pediatric, adult-only diseases, we do have a couple assets that are adults as well, so we can incorporate any learnings from that. I think mostly our concern right now is the pediatric population and the concerns with running those FIH studies in young children.
Yeah, but I think I'd just say that the playbook we envision will explain that the considerations for pediatric AAV gene therapy are different in some ways than for adults, and that will be clear.
Yeah, and I imagine a little different as well for a developmental gene that then you may have lived to adulthood versus an adult onset gene that's going to then trigger degeneration and death over time, just as in terms of the benefit risk as well as the timing and what types of benefits might be. Thank you.
Hi, Zach Walls, UCB. Thank you so much for your presentations. Dr. Brooks, I was struck by something you said about one of the goals of the AMP being to change or influence the considerations of what makes a commercial disease. And I'm thinking in particular of certain instances where a sponsor has gotten marketing approval but not had alignment with payers. So is a conversation with payers within the scope of the AMP, or how are you addressing the considerations of what is a commercial disease?
Yeah, I don't think we've thought about it in terms of discussions with payers. I think what we're envisioning is if a company is thinking about, do I want to develop an AAV for this disease, how much is it going to cost me, how long is it going to take versus the potential profit I can make. If we could come up with a streamlined process so that the path to getting there is faster and less expensive, that might change the decision as to whether it's worth going forward. I think that the issue about the payers is a very important one, but generally that's beyond the scope of the BGTC.
Thank you.
Hey, Stephen Gray from UT Southwestern. First of all, I mean, the BGTC, I'm involved with this, very excited about it. It's going to be very useful to the community. This is going to be an incredible resource. It's also very ambitious. But, you know, it's really focused on the preclinical to end of phase 1/2 aspect, you know, the translation, the first in human. And I think that this is kind of touching the last question. You've got the downstream components of that. What happens after these 5 or 6 years? What happens after a disease goes through a phase 1/2 trial and hopefully gets very positive clinical data? You know, are there concerted efforts within the BGTC that are going to try to address the instances where a commercial entity won't step in to pick up those diseases at the end of a positive early phase clinical trial, you know, just because of patient numbers and economics? I'm just thinking about, the long game and the long view to solving this bigger problem of bringing these treatments to patients.
Yeah, Steve, so I think the best case scenario is that we will do some of these, the clinical data will come out really well, and companies will say, that's a great asset, I want to pick it up and commercialize it. We also realize that's not necessarily going to be the case, and I think we've seen in other gene therapies, the commercialization is really a challenge. I mean, I think about, you know, ADA SCID, that I will say is legitimately a cure for a rare disease. It's been approved in Europe, and currently no one has found a way to market it. So I think there's a challenge here that really goes beyond the BGTC in terms of, can we identify different entities that are going to be able to step up and market drugs? And we have had some discussions about that and whether there's a role for the FNIH in there, but I would say those discussions are ongoing. Okay, I guess we're out of time, but thank you everybody for the attention and questions.